Today, I'm going to show you how to take environmental samples, and we'll also learn the proper way to label plates. So the first thing we're going to do is just take an environmental sample from somewhere on college campus. And we're going to use what we see here, a nutrient agar plate. Now when we look at a nutrient agar plate, this has agar in it. And agar is food that we use to grow bacteria with. When you label a plate, you always label it on the bottom side, the side with the agar. And the reason you do that is twofold. First, if you look carefully at the lid here, you can see that there's condensation on it. If we were to keep the plates like this, the condensation could fall down into the media and spread the bacteria everywhere. Also, if you look at the plate, you'll see that there's a little lip here. This prevents if any dust or mold or mildew falls down into this little rim. When you flip it over to open it, it all falls out. And so this helps keep the plate sterile or keep it free from contamination. So you always store a plate with the agar side up, which means that you're going to write the label on that side. So I'm going to go ahead and label it, and you always write your initials. My initials would be AAM, and I'm going to put my lab section, section 21. I'm going to put the date, whatever your date is that you're taking the sample. I'm going to put the type of agar. Now this is nutrient agar. So we would normally abbreviate that as NA. And I'm going to go ahead and zoom in a little bit so we can see it better. I'm going to put down 37 degrees Celsius because that is the temperature at which I'm going to be incubating, and I will write the name of the organism. Now, I don't know the name of the organism in this case because I'm sampling an unknown. So I think I'll sample the bottom of my shoe. So I'm going to write bottom of shoe. So notice I'm labeling those around the edge of my plate. Now, to take my sample, I'm going to be using this sterile swab. The sterile swab comes in a pack of 2, and it has an end that you can peel open. So you'll see when you peel it open, here's our 2 swabs. I'm only going to be using 1. Normally, when we deal with bacteria, you will always wear gloves. But because I don't have any bacteria on the plate yet, and I'm taking a sample of a shoe, which is a normal thing that I don't wear gloves to put on, I'm not going to wear gloves to take this sample. But I have some students that have taken samples of cancer cells, taking samples of cow with mastitis, skunk scent glands, all sorts of things. If I were doing something like that, I would probably wear gloves. But because the bottom of my shoe is dry, I need to dampen the swab first. So I'm going to use just tap water, and I'm going to spray a little bit of tap water on my cotton swab. So now it's wet. And I'm going to take this cotton swab and rub it against the bottom of my shoe. And you can see that my swab has this sample on it. Now, when I'm applying it to the plate, I'm not going to just rub it, because if I do that, I might be missing the place where my sample is. Instead, I'm going to take my cotton swab and I'm going to roll it across the surface of the plate. And by rolling it, I'm guaranteed that my sample is going to be put onto the plate. Now, agar has a consistency kind of like really soft Jell-O. So I am just gently rolling this on the surface. If I were to press too hard, like this, you can see that it's cracked the agar. So you do it very gently. And when I'm done, I'm going to go ahead and put this cotton swab in my biohazard container. Now notice we have another plate here. This plate is called a quad plate. If you were doing this in person, what you would do is you would take one of these quad plates and you would take 4 sterile cotton swabs and you would go home and choose 4 different places in your home environment that you would use to take samples from. And people can be very creative with what they sample. And then we would bring this plate back and put it in the incubator. So I'm going to go ahead and put this plate in the incubator, and I'll do some samples and we'll put that plate in the incubator. And next week we'll see what grows. In addition, I have 2 test tubes. I'm going to do what's called a thumb inocula. Now, inoculate means to put bacteria into something. So right now, there's no bacteria in these broths. I'm going to go ahead and put bacteria in them. And I'm going to do one with my unwashed thumb, and then one with my washed thumb. Now, to distinguish those, I have a roll of tape here, and I'm going to write the same things on my tape. I'm going to write my initials, which for me would be AAM, my lab section, which is 21. I'm going to write the date, which I took my sample. This is nutrient broth. Broth is a liquid, agar is a solid, which we would normally abbreviate as NB. I'm going to be incubating this at 37 degrees, and this is my unwashed thumb. So I'm going to go ahead and put that on my test tube. And now I'll take the lid off, and I'm going to do my unwashed thumb, and just 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And then I'll cap that off, and I'll put that in the incubator. I'm going to do the same thing with my other tube. I will go ahead and label it up, wash my thumb, and do the same thing, 10 count, and put that in the incubator. And when we come back next week, we'll go ahead and see what we've got. All right, take care. I'll see you then.